All right, so webinar 7 of 7, this is our imposter workshop. So we're going to go through some lookalikes, some very common lookalikes that a lot of you may struggle a bit with, or maybe not you who are on this call, but I think agents in general and stakeholders in general. So what's coming into our diagnostic lab, what emails are coming into my, my computer, and even some of the other specialists. Things that are easily confused with disease, and we wonder what the heck's happening. Also remember that herbaceous plants usually react differently than woody plants. And then deciduous plants will really act differently than evergreen plants. So depending on the actual plant and how the plant responds to an environmental stressor is really important. And knowing the plant, knowing how it reacts. For instance, peach. A lot of times you'll see the oozing out of a peach, but that doesn't mean it's a certain kind of canker or a certain kind of insect. That's just the reaction of the plant. So we have to kind of know those things. For those of you who work like a broader crop range, that's where it gets really hard. Ornamentals seem to be the hardest because we've always got new cultivars and new species and just new things all the time. And even, you know, whatever used to be on HGTV or in Martha Stewart's magazine always seemed to be what people decided to plant. And then, of course, we were stuck not knowing anything about them. And we kind of struggled to kind of keep up with the industry. Row crops are much easier than that. You pretty much have your standards and you can kind of learn it a little bit better. But that's never to say, though, that there's something different that can happen. A really wet year this year, a few years ago, really dry years. Some of these late freezes and frosts putting stresses on plants with a frost cracking. So all of those things will manifest into stresses, and stresses then either invite secondary pathogens, so we can have secondary disease, or they can just bring in things that you'll see today that I have. Dry seasons, we see a lot of spider mites. Wet seasons, we see things like root rots, or just you know, just wet feet, if you will. So let's start a little bit. I'm going to kind of be all over the board. I'm going to try to focus a little bit, and then I want to go through just a few pictures. So if anybody wants to stop me, feel free to do so. I'm looking at the questions. Okay, it's not a question. All right, so one thing I want to show everybody, you know, we discuss all the time on what is normal. And knowing what is normal is absolutely critical when we go through and try to diagnose. So at the beginning of this whole series, we talked about making that field visit, looking for patterns, all of those things. We hope to do those exercises for the September practicum. But knowing what a healthy plant looks like, and that seems really kind of obvious, but this is one that really gets me. This is a pine. Look at the curling. This is not herbicide damage, guys. This is normal. So how do you, okay, my camera's focusing. So how do you know what's wrong with this pine when indeed this is normal? So this is an extreme example. I'm very aware of that. But what is normal? We even have a lot of hemp cultivars. One of the parents of a series of CBD hemp has a lot of chimeras in it. And they just kind of pop up randomly, really kind of virusy looking as well. So knowing what's normal, but this kind of wonky thing, again, is extreme. But getting out there, especially if you're new or if it's a new crop for you. We've talked a lot about, and for those who took my diagnostics class a few summers ago, and maybe I'll try to bring in for our practicum, what has hairy and fuzzy leaves, like where are those trichomes really downy, and you know, what's the difference between a hairy grape leaf and downy mildew on the back of a leaf. Sometimes we question because it really is hard. So if you're not sure, get healthy tissue, look at it under the scope wherever you need to look at it, and know. So I really wanted to bring that up, know what normal is. And sometimes it's not quite what you would expect, and getting down and dirty just at the same level you would for a pathogen. Okay, another very common question we get, especially I get personally through email, is what we call transplant shock. Transplant shock is kind of a local term. I had never heard that term before until I moved here. The concept is all the same, is reactions, and it's just a group of symptoms that can develop on especially woody plants within the first 3 to 5 years of planting. And you can have a shorter version of that with an herbaceous plant. But what happens when you first plant a plant? All the things that plant needs. You've got a reduced root system. You've got a change in exposure, a change in temperatures, change in soils. And when you do, how is that plant going to react? And what have you lacked in giving that plant? So you can have everything from insufficient water to the hole not being big enough or being planted too deep. All of those things. We have a transplant shock hub, by the way, out there, but it just kind of gives you a range of all the things that can happen. What did we do? Moving from, let's say, the nursery into the landscape. What did we do? What has that plant lacked? And where do we drop the ball? I have one here from my yard, and I will call this transplant shock. So this is a dogwood. It's about 3 years old. This one is green sleeves, but look at the marginal leaf scorch. So this is absolutely transplant shock because this tree has not yet established enough. It hasn't developed a root system that is large enough and extensive enough to take up the water and nutrients it needs. By the way, there's a hackberry, a really large hackberry across the fence. So it's a very dry area. It's dry shade where it is, and I didn't have enough irrigation picks. I do drip irrigation. I didn't have enough irrigation picks in that bed, and this tree is very dry. So remember, we talked several times about marginal leaf scorch. So marginal leaf scorch is really initiated either in the tip or sometimes around the edges. And that's usually an indication that there's insufficient water and nutrient uptake, usually water uptake. So transplant shock, even remember we talked about bacterial leaf scorch, and it initiated a marginal leaf scorch because of clogged xylem tissue. But this one is just dryness, even with all the rain. This poor little dogwood needs me to step up and add some more irrigation picks there. Do a better job. So that's just one example of transplant shock. Planting too deep, looking for things like root flares. We talk a lot about that in a lot of sessions that you see me. But really important is that we understand what a plant needs, how a plant grows and establishes, and asking the right questions, like what is the big picture? And we talked about that in sessions 1, a little bit in session 2, but mostly in session 1, we talked a lot about looking at the whole area. Where did the plant come from? What is the site actually like? So this we may or may not consider transplant shock. Doesn't matter when, but this is a nutrition issue. Nutrient deficiencies are very common. This is another dogwood out of my yard, as a matter of fact. It was in a pot for too long. It was just a little seedling. I think it was like an Arbor Day tree or something, and I don't throw away trees. So it was just hanging out in a pot. And but deficiencies, these are very clear and very obvious. But if you saw the tree itself, the new growth looked like this. But the older growth, is it clear, Kim? But the older growth looked fine. And that's kind of the tricky part is as an agent, you don't always see the whole plant. You only see what the stakeholder brings into your office. So what did the rest of this plant look like? What is the story here? I'm guilty. I'm telling you that there's a, there's a bad treatment that went into this, but a lot of times our stakeholders won't say that, and they just say it's looking bad and I don't know what happened. It was just suddenly, and this did kind of pop up suddenly, or was it sudden because I wasn't paying attention and I suddenly noticed it? But anyway, so for knowing, again, backing up, looking at the whole plant, looking at the whole site, how long has this tree been here? What's going on? But again, we can do these kinds of things, and we can really forget that that we have a bigger story, right? So, and again, that one is really obvious. Some are not quite so obvious. So here's another nutrient deficiency. I'm going to try to break this up for you. This is a nutrient deficiency. This is a deciduous magnolia. And let's try to come a little bit closer. So you'll see just kind of off color. You see these black dots. And this is kind of the kicker. Some of you have probably already seen the sample. I bring it places. Look at this. This really looks like a fungal leaf spot. And it's a new growth, or spring's new growth. And I know this tree. I've been watching this tree for years. This tree, so the pH in this area, this is a tree right outside of my window. This tree has concrete all around it. A little courtyard was built. The pH is high. And magnolia, we know about magnolia, they like a lower pH. So a lot of nutrients are unavailable when the pH is not within range of whatever the plant requires. And so a nutrient deficiency begins, a little bit of chlorosis in the spring, very obvious nutrient deficiency in the spring, but that weak tissue becomes necrotic as the season goes on. And this just really looks like a disease. So it's in certain parts of the tree. It's in the new growth in the springtime. And you see the growth scar is not very far. This poor tree is suffering, but this is a mature tree. This is a 20-year-old tree, and it's pretty large. It's probably 25 feet tall, and it really has a nutrient deficiency that looks otherwise. So be careful when you try to figure these things out, asking all the questions that we ask, and it's sometimes frustrating, but we know. So I can find a few fungal leaf spots. I can find spider mites. I can find all these things, but in reality, our issue here, see, we've got some kind of insect issues. We got just some mechanical wounds because people walk by and scrape their bags and purses against the tree. But this is a nutrient deficiency that manifests itself because this is a really rigid leaf. So instead of turning yellow and falling, this is kind of what happens. So knowing a lot, and sometimes we just don't know. Had I not been watching this tree and out of curiosity, we kind of tested it, I would have never known that that's what was going on here. So as agents, you may never know. You may never be able to put the time in to explain something like this. And speaking of slow, and I'm talking woody today, I realize that. But when we talk about unthriftiness or unhealthiness, thinning canopies, slow dieback, when we ask ourselves what that bigger picture is, we know that a lot of times it happens really slowly, and if it happens that slowly, it's rarely a disease. So thinning canopy is something to always look at, and a lot of people don't realize right on. And if you've got that issue, then you could track back, and I know a couple of you, I've worked with you on certain issues, and we can make guesses and we just really can't figure it out, or we can't invest the resources for extensive testing when in reality the plant is declining anyway. So I'm trying to figure out what's primary and what's secondary. Any questions on general dieback? So some terms, remember some terms we used early. Flagging, that's when one branch is dying or dead and all the surrounding ones are healthy. So flagging is usually an indication of some kind of canker damage or insect damage or girdling of some sort. So it's not a whole plant problem. If we've got dieback, thinning canopy, stunting, remember we looked at the terminal growth scar. Did we look at terminal growth scars in here? No. Okay. Uh-oh. My office is really so messy. I don't know if I have that. Okay. I'll take a break and I'll send that at least as a photo. But looking for a terminal growth scar, darn it, I am so sorry. Okay, there might be one there. Looking at a terminal growth scar will let you determine how much growth the tree got that year. So what is the new growth this year? And sometimes you can look and see just nice soft green tissue, semi-woody, or you can see color of the new growth. It really looks different. Other times you really have to go back to that terminal growth scar. You can see those little rings. I'll show that to you, but another indication is lichens. Lichens are not feeding off of the tree tissue at all, but lichens, they're kind of air plants, if you will. So they're kind of hanging out. They're hanging out on the bark. They're utilizing any free organic matter, whether it's blowing around in the canopy or in the air, or they're utilizing some of it from bark. Different types of lichens act different ways, but they're not feeding on the tree, if you will. But as a tree grows, think about that it sloughs off its bark, right? So bark sloughs off as the tree expands. So if a tree is expanding quickly and growing quickly, it's sloughing off that bark fast enough that lichens can't really establish in large numbers. If they're not growing sufficiently, that bark is not sloughing off, so lichens are able to stick around and to populate much more. So when we look at a tree and we see a lot of lichens, that's one consideration. Is this tree growing fast enough or not growing fast enough to slough off that bark? Secondly, some lichens will consume that dead tissue. So they're hanging out there and they've got an all-you-can-eat buffet of dead plant tissue if that plant is declining. So if you see a lot of lichens, again, you're backing yourself up and asking, what is this tree health? What is the health of this tree? What can we do to improve the health of this tree? So it's not specifically disease, but your lichens are not a causal agent. I'll tell you the story. My grandmother used to go out. She had huge azaleas, and she would go out with a butter knife and bleach, and she would scrape lichens off to improve the health of her azaleas. So the things that we do sometimes we don't really realize, or the things that I watched her do it, and I knew that was wrong back when I was a kid. But hey. All right, so we talked a little bit about dyeing back. So this is dieback. So this is a beech, but look, we've got kind of some tip dieback. We've got really thinning branches. Some twigs have no leaves. Some have just a few leaves. So this would be kind of a dieback or a thinning canopy. So not dead. And I am not wearing my glasses, guys, to find a terminal growth scar. Let's see. I'm going to break off here and see if I can get this one out. I'll show you some better pictures. So I'm looking right here. You'll see kind of some rings. So tell me if you can see. No. All right. I will put that on pause. When I crank up the dyno light, I will show you the terminal growth scars. I'll put them here for you. It's something you really need to see, especially if you're dealing with woody plants. Really important, and it's something you could always show your stakeholder and say, look, this plant is not growing well, this plant is unhealthy. And then you can start working through soil tests. A soil test will always tell you something about what's going on underground. Maybe you can't correct it, but you definitely know how to replant or how to treat what comes next. All right, so that's kind of all I have for I have another abiotic issue. So we'll keep going. Any questions? Okay, so this is another magnolia that's outside of our door. This one has good terminal growth scars. I'll show you these. But this one has, I'll call this mechanical damage. And you'll see, look at these cankers. Let it focus on for us here. No, not wanting to focus. All right, technical difficulties, section 7. So there are several. Not cooperating. All right, I guess I'm going to have to flip my dyno light on just to do this. So this is rubbing. All this rubbing. This branch was right at the height of the roll bar on the mower, and so we see this a lot. So when we see this, so the difference between mechanical damage and a fungal canker is usually depth. What is the depth here? If you get into the cambium and you've got a really clean cut, we can always cut back here and we're not going to see that gradient of necrotic tissue. But this is really clean. There's no rot, there's no discoloration, and we're down into the cambium. We're down past the cambium, and that is when we suspect mechanical damage and start looking at what the location is. The location is usually down at ground level, a few inches higher, usually a mower deck, or it can be as high as, say, the roll cage or the roll bar of a mower or a tractor. And this one is full of it. So I will show you that one. I'll switch on my dyno light and show you kind of what that looks like. And the mechanical damage can come in lots of shapes and forms. We'll probably see a lot of that. Also string trimmer and mower damage around bases of trees. I've got some pictures. We'll flip through some pictures as well. So mechanical damage, look for that. Look for stresses that way. Any questions? All right. Another one that we kind of need to cover is sunburn. So, oh, I'll show you. I'll show you my tree. For those of you who are not my Facebook friends, this is my tree. You see the included bark. Let's see if I can get it in there for you. See the included bark? We had a windstorm a few weeks ago, and the tree split. So, really pretty rough, but this, here we go. See this included bark. So we have very weak branch angles. I knew better. This is a black locust, the purple robe cultivar, but we've got lots of included bark, and it just went down in a storm. Okay, so I wanted to kind of bring that one. This is something, I think it's a really good example that even those of us who know better make really bad mistakes. And I know I show you a lot of my bad mistakes, but this is absolutely one. But this black locust, I'll tell you a little bit about. The tree is, it's a very fast grower. And I put this tree on the southwest side of my house, in my front yard, and it grew really fast. I had a 20-foot tree by 15-foot diameter canopy in 3 years. And suddenly I switch out all the plants in my front yard because I had shade. And so I lost my tree. Well, maybe it was a month ago. I lost my tree. And suddenly I had sun again. So I have all of these shade plants in a sunny spot. And this is an example. This is sunburn. So this is a variegated. This is a Brunnera. Variegated. So that's kind of what it's supposed to look like, but very dull. You see marginal leaf scorch. And you also see, look at the way this guy's spotted. So this is not your typical, this is not what you would think of as sunburn. You can look through these spots. There are no fungal structures, not at this point anyway. No fungal structures, there's no streaming. It looks bacterial from a distance. If it was bacterial though, there would be water soaking and you could stream it. But this is very dry. It's really crispy. I don't know if you can hear it. It's really crispy. It's really having a rough time. But just take a look at this poor leaf. So just take a look at what's going on. You've got some whitening. So that's the cuticle that's kind of flaking off. So sunburn really will manifest itself differently in different plants. You have to know that history. So I could have come into my county agent's office and said, it just suddenly happened. It was healthy and it suddenly happened. I kind of did, but I went from shade to instant sun on the southwest side of my house. So thinking about, and these things can happen. These things can happen in a soybean field. If we've had a couple of cloudy, rainy weeks and suddenly we have a heat wave and sun, we can have that same scorching, that same sunburn in situations that you just don't usually see it, and you have to back out and think about what was the weather. Some more sunburn. Here are my hostas. These all come from my yard. So my hostas. So we see a couple of little leaf spots, but marginal leaf burn completely, and just really suffering. And then bleaching. So look at the white. You can kind of see there's kind of some bleaching, and this is a variegated hosta, so it's kind of hard to tell, but there's bleaching in here. And if you're looking at sunburn, I'll give you a hint on how to look at sunburn. So the plant, however the plant sits, think that sunburn is going to hit the section of the plant that's facing upwards or facing the sun. So if you've got a plant, you've got some leaves that are higher than others, you would see the sunburn in that pattern that the sun is hitting. And then you would see the lower ones not. But sometimes you'll see a leaf that's really curled and you'll see that sunburn just kind of coming across almost like a line. So think about how the plant is positioned. When samples come to your office, you don't really see how it is positioned. You can guess, you can ask questions, and a lot of times you can ask for photos. If you can't make every, and you cannot make every field visit, especially with homeowners, but trying to figure out what the placement is and trying to look for patterns. Questions there? All right, here's another sunburn. This is Japanese maple. Look how hard. So this is the filigree leaf. It's not going to cooperate. And there is some marginal leaf scorch in these little tiny leaves. Let's see if I can get you this. No. We're eventually going to figure out how to focus in. It's focusing on me somehow. So we've got some marginal leaf scorch, but just think about the shape of these leaves and how a marginal leaf scorch looks one way on a very wide hosta leaf and another way on this very narrow maple leaf. So just think about that. We're not going to go into any details on it, but think about that. Any other questions? Any questions on abiotics or environmental symptoms? All right. So some other things that we've really been struggling with lately are spider mites and a lot of scales. This has been somehow it's been a rainy summer, but I'm seeing more spider mites or getting more complaints about spider mites. I am not an entomologist. I'm not going to talk about managing spider mites or any insects or arthropods at all. We've got some agents, as a matter of fact, who have their degrees in entomology, and they can tell you much more. I just want to kind of point out where they can be mistaken as disease or some kind of suffering. So I'm going to show you first some scale. Look at this nutrient deficiency. Look at that, at how horrible undersides of the leaves we've got scale. Those are scales. So this is inkberry. So really looking and investigating, this is very extreme. You don't have to investigate really hard, but this didn't happen overnight. There obviously has been a scale problem that I know, this is mine as well, I know this is a male of the inkberry, and it's my only male because the others are females, and it blooms just enough to get berries in the females, but I didn't really want it to grow that big. It kind of looked this way, but it bloomed enough to give me pollination. But this did not happen overnight. This has obviously been going on, and I did not recognize it. I did not look for it. I did not care. And now I've got a real problem. As a matter of fact, the scale are spreading. They're spreading to a lot of things around it. This is a PG hydrangea. You can see there's that scale starting to come in onto this hydrangea. It's a little bit wilted. I cut it this morning, but you've got the scale everywhere. So that's going to be the next problem. So I've really got to address this, and I let it go. And again, if I let it go and I know better, think about sometimes where our stakeholders don't quite know. But these things don't happen overnight. They happen pretty slowly, and I never saw it. I never paid attention, but I was at eye level with it many, many times every year. So neglect gets us into trouble sometimes. I'll show you an obvious one, and I know everybody on this call knows I won't say it, but Japanese beetle. So we all have choice words for Japanese beetle, right? So this was on a crabapple right outside the door. We all have Japanese beetle damage on lots and lots of different things. But we even got a couple of samples, Japanese beetle damage samples, in the diagnostic lab. And I know sometimes you guys are out of the office, or some counties don't have agents, and the staff just kind of packages things up. But Plant Disease Diagnostic Lab does not help anyone with this. But the Japanese beetle, just kind of knowing what's going on, following KPN, there are a lot of articles on those kinds of things to help your stakeholders with that. Any other questions? All right, moving on. Spider mites. Spider mites often are mistaken for disease. This is Alberta spruce. I guess my shirt is just confusing this this camera. So spider mites on Alberta spruce. Most of the time, Alberta spruce suffers from spider mites and not any disease per se. They're not happy here. Mites love the dense canopies, but large holes, brown needles from the inside, and I'll turn my microscope on in a second. Spider mites galore. Spider mites have manifested themselves in lots of ways this year that I had not seen. This is spider mite damage as well. So this is Veronica. So they came into the flowers. They just destroyed the flowers. So we'll see stippling on the leaves a little bit, but not as much as you would expect. And the webs were here. So, okay, I have lots of those samples. But suffering from spider mites in ways that I did not expect. All right, spider mite damage also, a lot of bronzing. So 2 symptoms on spider mites. One is the stippling, where the piercing, sucking mouth parts have probed into individual cells, and those cells turn white. Another is bronzing. Okay. Let's see if we can get the color here. Another is bronzing. So turning bronze this way, see in between the leaf veins, you'll see the tissue is kind of turning that brownish bronze. Very classic of spider mites. But it can also be the discoloration between the veins, like a chlorosis, can be the result of something like a root rot or a large canker in the trunk of a tree or anything like that. Bronzing, though, is typical of spider mites. Think, though, that just when you think you're sure, you might not be. Okay, so seeing that bronzing there. It's not brown. It's really bronze. Check it. No. So that color difference. I'll try to get some photos here for you so that you can really see that difference. Questions on spider mites? Okay, so I'm going to switch to my Dynolite. I want to show you stippling and how that's different than some of the diebacks that we see with our diseases. All right, so this is celery here. This is, you see the spider mite. They're crawling in there. Spider mites, you see frass, you see some webbing. This is, let's see if I can get these unfolded. So you see the spider mites and crawling around under there. 2 spotted spider mites, see the 2 spots. Try to lower if I don't shake it. And there we go. There are the spider mites crawling around. You can get it much closer than this, but they're crawling around. They're not dead. I sprayed horticultural oil. You see the frass under there. The round globe circles are the eggs. So there's lots of eggs. There's lots of frass. There's lots of spider mites under there. So all of those black dots you see are the 2 spots that are on the backs of the mites. Nope, I thought I would get closer without not making too much of a mess here. But look for spider mites is my point here. So this is the celery. I'm going to show you stippling. This is a tomato. That's a close-up of stippling. Rick Besson put a, I think KPN this week or last week, there was an article on stippling. Or on spider mites, and he described what stippling was and what it looked like. But this is it, and this is pretty bad. These are my tomatoes, by the way. So each of the white spots is the sucking and emptying of those cells. Pick up real quick. So this is a really bad, case of spider mite damage. And on the back side, what do you get? you get the mites and the webbing and the frass. So you can always see those moving. I was showing you earlier my celery, sad celery. There it is. How not to grow vegetables. Ask Nicole. Come to see my yard. So, and then you've got these tomatoes. This is what happens when I wait too late to spray. You go from the stippling to this, and no fruit. So this is just shriveled up. Shame on me. So I have a real spider mite issue. Any questions on spider mites? Besides the fact that they're crawling all over my office now. All right, so I'm going to backtrack real quick while I have the dino light on, if everybody's okay with that, and show you some terminal leaf scars. Here we go. See the little rings? See these little groups of rings? Those are terminal growth scars. And if I back up, I want to show you just how sharp these are. This is that tree that was suffering earlier. So not a lot of growth. Look at the distance. Look at the difference between scars. So we're not getting a whole lot of growth here. This tree is really suffering. This is that beech that I was showing you earlier. Okay. Let's see a larger one. See if we can do this. This was the mechanical canker, by the way. See kind of the shredding. This is the mechanical damage. There's a scar. Let's see if I can break this up for you. Get it under the scope a little bit better. So the scar is going to form here. Okay, so we haven't gotten much growth. We haven't gotten much growth at all this year. This is another suffering plant. It's in a really bad spot. Questions? All right. A few more insects. I'm going to switch off. Let's see. Let me see if I can do it from here. Another one we see a lot of and is not easily recognized is bagworms. Bagworms, we see a lot of bagworms on conifers. And there it is, the bag forming. The bag forming, so the arthropod will, I can't remember the details, I'm sorry, again, I'm not an entomologist, I should have looked this up. So there is a bag here. Try to cut it open. Oh, wow, I'm making a mess, guys. And inside, you will always find a little worm or caterpillar. Okay, I can't pull it apart. So opening inside, so it is truly a bag. Luckily, she's gone. We didn't see her today. So bagworms, really common. I'm going to switch back out. I'm going to toggle back out. Show you. Let's see, stop share. Here we go. Okay, so bagworms, this is a conifer. You'll see the bags. See the bags happening there. Here's another bag. All right. Once it's so severe, you see a lot of death. There are two bags there. So in a lot of conifers, look for bagworms and mites. But this really would look like a tip blight if you didn't look closely. And sometimes they're hard to see. So not always a tip blight in your junipers or your arborvitae. A lot of times you can have bagworms. Bagworms, just like anything else, they don't suddenly appear. You'll have a problem that gets worse year after year. And when there's an environmental stressor, the plant is just so weak from these kinds of issues. So cutting them back and it just really knowing your plants. Really important. Okay, so what time is it? It's a little bit after 10. That's really all kind of I have. If anybody, oh, let's see, wait, I see questions. Can aphids cause stippling too? Yes, Kathy, they can absolutely cause stippling. Any insect that with piercing, sucking mouth parts will stipple. And that's just a symptom. Remember, with plant diseases, we talk about symptoms are not signs. A symptom is just a reaction by the plant. Same with your insects. So you can have symptoms, but you have to identify that that insect, and your entomologist is the one that's really going to help you. But you've got to search, you've got to get those samples, and your Dynolite can identify a lot of these things in advance. And then you can send pictures to an entomologist if you need help with that. You also have some praying mantis. Where did I had some praying mantis? Did I have some praying mantis? One year, the diagnostic lab had a praying mantis egg sac, and they brought them to so the diagnostic lab. On the scale, did I really? And it's in my bag. But anyway, somebody sent praying mantis eggs into the diagnostic lab, and over the weekend they hatched, and there were a bunch of them all over the diagnostic lab. So who is that? Amy? Tell about cicada damage. Looks like the young twigs of woody plants. Yes, cicada will lay eggs in a young branch. You see it a lot in nursery stock. Okay, we're distracted by praying mantis now. I'm going to give them to Kim. Kim is on the mission to find him. But cicada will cut into a branch and lay their eggs inside of a young tender branch. So cicada damage will usually be on the underneath of a twig. It's usually a young twig, and you'll see a very clean linear cut, and usually there will be a bunch of them. And you won't see, again, you won't see the rot. So taking your blade and cutting into a canker, and I can do this under the Dynolite if you need me to, but cutting into what we would call a canker, and as you see here, this damage, and a cicada damage will be the same way, is that when I cut into it, it's still green. You don't have that gradient of decay like we do with our pathogens. Does that make sense? He was so small. Amy said he was very small. He is in trouble because I never saw him. He's not there anymore. Okay, I'm going to be on the lookout for him so I can send him back, rescue him so he won't get stuck in here all weekend, right? Any other questions? Thank you for that. I love praying mantis. Anything else? And I know, again, we see in frost cracking. Kathy, frost cracking, I thought we talked about that. I guess we didn't. Frost cracking will usually occur late spring. We also have a publication, by the way. So frost and freeze cracking occurs in late spring. And this is what happens in the springtime when we start warming up, then it'll get cold again. So once our plants reach their chilling hours and we get a warm spell, the plant starts breaking dormancy, and you get that fast flowing of the xylem tissue. So basically the sap is up. If you've heard that term, the sap is down in the fall and the sap is up in the spring. So what happens is the plant starts actively pumping water through its vesicles. And when we get a late freeze, suddenly we have all of these, this water in the cells. And when we get these very low freezes, it will freeze them or crack them. Yes, frost and freeze cracking are the same thing. We use them interchangeably. Sun scald can be used two ways. Sun scald we usually refer to sometimes as that frost and freeze cracking because they kind of go together. We get the breaking of dormancy, and we get an actively flowing xylem system. Then we get the freeze that comes in. and that water will freeze in the cells, and it will burst those cells, and it'll freeze linearly. And usually that bursting will happen like the next morning when the sun comes in and hits it. So you've got warm, breaking of dormancy, then you've got a hard freeze, and you've got the sun that comes up in the morning and warms that tissue up really fast. Just think like when a pipe bursts. It's kind of the same thing. So you get that linear crack. That'll usually happen on the southwest side. If it's kind of an afternoon, late summer. But if it's happening, that frost freeze, it'll usually happen more in the east. So where you get it, where it's really cold, and then that east sun comes in and it hits it. It'll usually be a linear crack, and it'll usually be plants. We can lump that into transplant shock, right? Because think about thin-skinned trees. They're usually young trees, maple, apple. Some of these, maple are really notorious for cracking. Because there's that really thin skin before the bark starts getting scaly. Usually these trees are in a nursery. They're very close together. The sun really never hits them. We take them out. We put them in a scorching landscape. And then we could get that afternoon beating sun in the summertime. That's what we would probably call sun scald a little bit more. But when we have that winter cracking, that's still the thin skin trees. So I know I'm all over the place with that. Let's see some crepe myrtle yesterday, and they had large trunks, deep cracks in the base of some of the trunks. Linear cracks. Wow, I didn't know about crepe myrtle. They were dead on the trunks with vertical cracks. It may very well have been. I doubt we would see crepe myrtle or tropical. I doubt we would see that summertime sun scald. It may have been that frost or freeze cracking. But again, look at what side it's on. If it's on the west side, it's scorching. And it just really was heat and scorch and just a shock. Again, coming out of a nursery, coming out of a shaded nursery area, and then being put into the sun and the sun beating it down. As opposed to the frost freeze cracking that when the sun hits it on the east side in the morning, it'll crack it. So look at what side that's on, Kathy. And really think about when you put, especially a maple, into a landscape. We always talk about wrapping the trunks, or that's why painting the trunks white. I know it's hideous, but never wrapping in the black perforated pipe, okay? Because that's black, and it's just building up heat. It's heating it up. So if you're going to wrap, Bill Fountain recommends window screen and just stapling it on, because that lets air flow. It's very loose. And when you staple it, it moves a lot, but then in a couple of years, those staples will kind of rust away. And then even if you don't take it off, it kind of fixes itself. I want to share, speaking of, some pictures. Nope, sorry. You don't need to see my email. Let me find it, guys. All right, you should be seeing this. All right, let me try to share screen again. I have to show you some pictures. There it is. All right, are you seeing this? What are you seeing? I'm looking at, Kim has her laptop. She's kind of my proctor here. Is that just black? There's like black in front of it. Why is there black in front of it? Oh, sorry. Let me try something else. So now are you seeing? All right. We'll do it this way. I don't want to spend a bunch of time on this, but I did want to show you a couple of things. No? Oh, guys. Let's see. Let me try something else. Let me stop share and share something else. No. It's not giving me a choice. There it is. Okay. So this is the black perforated pipe here. Do you see my cursor? Yeah. Okay, so it's digging into this plant. The plant has grown around it, and it has really cut into the tree. So plastic does not decay, FYI. So you don't want to put anything like that around it. There's a paper tree wrap that you can use, but don't use the black because it builds up heat, and especially will break that dormancy and heat it up much sooner. Oh, this is a hemp sample. This is compaction at its best. So this is a very limited root system, and when we got a heat wave, the plant started really wilting. So that is compacted soil. Okay, this is right outside of my building. I kind of wanted to show you string trimmer damage. Look at where the mulch, it's been mulched and not really maintained, and so the mower just gets closer and closer, throws mulch up, but this is string trimmer damage, just girdling away at this crabapple. Oh, there are the hemp plants in our lab with this little restricted root system. There we go. Kathy, this would be sun-scald or frost freeze crack. I don't know which side that's on, honestly. I should know, but I don't. I think this is the west side. So you'll see kind of a linear cracking. This is an old crack, and it has made an attempt at callousing over. The pictures I have here. Okay, here was that hemp sample that with the compaction. Look at this root system. All right, I took this one back in my hometown. These were some American holly that were planted down a boulevard, but look, they didn't backfill properly. Look at this exposed root system. Is that amazing? So these plants, there was a dead one next to it, and it kind of bent down. You couldn't see it from the top. You couldn't see it from looking up, but just really bad practices. There's our hemp sample again, so you see where the hard pan hit and just a very limited root system. Well, this one was better than some of the others. Oh, so Nikki sent me this. Nikki, I'm using your picture. She sent this by email yesterday, and there was another issue. There was a hole. It's like a bleeding. But look at this driveway issue here. I'm sorry, I can't get this picture turned. I was supposed to turn it before I shared. But look at this driveway here. So think about what's going on with this tree. You've got compaction, of course, by this driveway. You've also got a limestone bed. So what could the pH be in this situation? Oaks are acid lovers. I think that's an oak. They're acid lovers, and with this limestone here, imagine what the pH is. So it was, like I said, it was another question. It was it looked like something bored into the trunk. But I'm going to keep this picture, Nikki, if it's okay with you. But just that whole scenario, there are a lot of questions here. What's happening next? And think about as this tree starts declining. So we know it's got a limited root system, right? Because there's no way that there's a healthy bed of feeder roots 12 inches below all of that limestone and all of that compaction. So what happens next is this tree is going to start declining, and it looks like this is some kind of public space. I don't know if it's like a park or something like that, but what are the dangers as this tree loses its root mass? And as it loses its root mass, there's nothing to anchor it. The feeder roots aren't taking water and nutrients up. You're starting to see a thinning canopy, kind of I bet the terminal growth scars are very close together. So just thinking about that. But this was a very interesting scenario. Okay, and this one came in to the diagnostic lab, and I wish I had the apple in my hand. I should have walked over this morning to get it. This is phytotoxicity from some kind of spray. We have the spray schedule, but maybe there was oil, a residual oil still in the tank, and maybe an incompatibility with 7 or something. But this is a phytotoxicity. This is not bacterial. This is not fungal. And it's not insect. So we've all looked through it, but it was on one side. So if you would see the apple itself, it was totally a spray pattern. One side of the apple got blasted, and you see it kind of gradients out from around it. So not the best picture, but think about, again, patterns. Look at this. This is a bad pruning cut. So what is this going to turn into? I thought that was interesting. Okay, there's our knotted root again. Okay, there's my tree in my front yard, guys. So you see the splitting, see what happened during that storm. I'm still upset over it, right? Okay, I'm gonna stop share. Any questions? Okay, I promised you I'd get you out of here early, and I have not, so apologies. So we're kind of done for the day. Kim is going to post the link for the quiz. The quiz this time is really giving us our feedback. This is our traditional extension survey. So we're going to ask you a lot of questions. It's an extensive survey. So again, thank you, because where is it? Is it over here? Sorry, guys. I know I opened it. Here we go. Okay, I'm sharing it. I'm sharing my screen again. There. Okay. So we have a lot of questions for you, and you have to answer each question before you can get the next question. There we go. So there's the quiz. All right, can you see everything? All right, I'm going to go to the next page. Remember, you go to the training website. If you go to the training website, all of our webinars are here. If you want to see the field practicum details, we're going to start building right here on this link. But right now, the webinar section. Today's quiz is here. Kim put the link in the chat box, though, as well. There are several parts to each question, and a lot of it is typing in comments. You have to type something to go to the next. So we're asking for a lot of information, and we need that information, so we need some feedback. How would we do this again? How are we going to do even shorter versions of this in the future? Do you like this? Do you not like this? Again, we gave you this option as a way to substitute the need to drive, for us to drive to you or for you to have to drive to us. And even though we do a lot of our sessions in multiple counties, when we do them, it's still hard. So do you want to see more of this kind of thing, you know, maybe 1 or 2 hours every summer? Do you want to see it during the summer? I know it's everybody's busy season, but that's when we have our samples that we can show. What do you want to see? So kind of give us some feedback. Let us know what you want, what worked, what didn't. We know the glitches with the technology, getting things focused in. So we realize that stuff, but we really need to rethink that and look for options for next time there. Any other questions? All right, so Kim has pasted the link in the chat box. For those of you, here's our insect crawling on the table now. I'm going to kind of end everybody, and I'm going to put that link back on for those who are using the video. You have 1 week. For those of you watching this video, you have 1 week from the time we post. Kim and I are both on vacation. Believe it or not, we're on vacation today. So we are not going to be pasting this or posting the video up today. We're going to wait until Monday, and then we're going to give everybody a week after that. Also, sorry, I completely forgot about this. Next Friday, August 31st, we're going to repeat fungi. If you've already gotten credit for fungi, you can't get credit again, but you can participate. We're going to have some different samples this time. There are just so many darn fungi. We're going to have to just give you some more examples. So you are welcome to do that. It is not going to affect your certificate program, but if you missed fungi and did not get credit for it, you can. Okay, does that make sense? So if you miss fungi, you can use this one as a substitute. You've already gotten fungi, you cannot use it as a CEU, but you can definitely come on to learn. We will also put it online as Fungi Part 2, and that way you can see just some different samples. And I know the last time I did it, I had so many here. I think it was really disjointed. So I'm going to try to focus in on just a few things and maybe work on some techniques. Yes, I'm going to record Fungi Part 2 as well. It's going to be just like all the other sessions. There will be a quiz. Everything will be there. If you don't need credit for it, you really don't have to answer them. You don't even have to put your name in the box. I'd like to keep track of how many people do it. I'd appreciate it if you did it, but you don't have to if you're not getting credit for it. Questions? That's a little bit kind of rambling. Okay, all right, goodbye everybody. Thank you all so much. I'm gonna toggle back to the link just for those who are recording, for those who are watching the recording. And remember, you have 1 week from today to complete the quiz. For those watching the video, you have 1 week from today to, 1 week from the day we post it. And then we're gonna close the quiz. Remember to register for your practicum sessions. The sessions close 2 weeks before the actual session. So the first one has closed. The second one in Marshall County closes today, and we have Lewis County and Taylor County that are still open, but they will be closing pretty soon. All right, I am going to toggle back to that link for those watching the video. If anybody needs anything from me, feel free to email. I will be out today through Tuesday, I'm sorry. So if you really need something, just let me know or ring my phone. And what is that?
I'll be back on Monday.
Okay, Kim will be back on Monday. I will be back on Wednesday. But we're very happy for anyone who needs anything. That's what we're here for. Okay, so I'm sharing back my screen for those who need the link. And again, have a good weekend, everybody. And congrats, you survived 7 webinars of unpredictability. So, all right, I'm going to stop recording. Thanks, guys. Bye.